Good afternoon and a very warm welcome to you all, and thank you for joining us for today's webinar entitled 2-Minute PCR is here, NextGen PCR, an entirely new ultra-fast technology for heating and cooling of samples. I'm Damien Doherty, the Editor-in-Chief of the Journal of Precision Medicine. I shall be moderating the webinar this afternoon. I'd encourage you all to send your questions to the speaker throughout the presentation using the function you'll see on the bottom of your screen. We will, of course, do our utmost to answer as many of these as we can during the Q&A session at the end. Okay, so until now, PCR instruments have relied on Peltier elements to heat and cool an aluminium or silver block accommodating 96 or 384 polypropylene tubes holding PCR mixes. Using this technology, PCR times for 30 cycles, 3 temperature reactions are usually between 50 and 150 minutes. Now, NextGen PCR is redefining fast. The thermocycler introduces a new way to heat and cool the samples instantly with virtually no ramp rates, losing no time getting the samples to the desired temperature. The user can go from melting to annealing in less than 0.1 second, and for 100 base pair fragment, total reaction time is as low as 2 minutes, with 700 base pairs being amplified in 10 minutes. Adding NextGen PCR into workflows results in higher sample throughput using optimized applications. More than 10 full plate experiments per hour become possible. Operated at capacity, this would mean 80 384 well plates per day, resulting in more than 30,000 data points per day, far beyond the capabilities of common thermocyclers. This webinar today is sponsored by Canon BioMedical, who are focused on empowering the biomedical research and healthcare communities by developing, manufacturing, and marketing innovative technologies and solutions. Canon BioMedical is the exclusive distributor of the NextGen PCR products, and you can learn more about those products that Canon BioMedical offers by visiting www.canon-biomedical.com. Okay, so without further ado, let me introduce our speaker today. Gert is Director of Molecular Biology Systems of Huis, Netherlands. He is the inventor of NextGen PCR and has a master's degree from Leiden University in Biology and Physics. After teaching physics in Curaçao, Gert returned to the Netherlands and embarked on a career as an entrepreneur and inventor in life sciences and medical device technology. During today's presentation, Gert will focus on the technology behind NextGen PCR. He'll pay special attention to publications detailing 2-minute PCR data and how NextGen PCR amplified all 29 fragments of the BRCA gene in less than 10 minutes for subsequent Sanger sequencing. So, Gert, I hand over to you. That the floor is yours.
Thank you very much, Damien. Well, everybody, good afternoon, or for many of you, good morning, or for some, maybe good evening. Welcome to this webinar. The first slide shows the machine as we are bringing it to the market. There's not a lot to be seen, of course, it's a closed box, but you can see there's a color screen to operate the machine. I will go into further detail about the functioning of the machine in later slides. So, just to put it in printing, my name is Gert, Director of Molecular Biology Systems in the Netherlands. And this webinar is about NextGen PCR technology. You can view more on our website, nextgenpcr.com, where there are also a number of downloads available, publications about 2-minute PCR and about the BRCA1 gene. Now, this is all about simple solutions. We all know that in mathematics, the simplest solution to a problem that you can find is the correct solution. And in life, the simplest solution that you can find to a problem is often the best solution. Simple doesn't mean easy, but it at least avoids complicating things. Now, this is a very simple thermocycler. It's the first one, and what I tried to depict here, it's a bit schematic, but the blue is a cool water bath at annealing temperature. The yellow is a middle temperature bath at extension temperature, and the red one is a hot water bath at denaturing temperature. And the first thermocycler was moving an Eppendorf tube between water baths powered by a human with a stopwatch in hand. And in this way, we were able in 1984 to perform PCRs in 2.5 to 3.5 hours. It was tedious work. You had to be there and you had to keep moving the tube that you see here in the extension water bath. And then somehow we went from this simple solution, from moving a tube with PCR mix between water baths, to something as complicated as heating up and cooling down a whole aluminium block, or silver block if you wanted to go a little bit faster. Many of you will recognize this as the core of an old MJ Research PCR machine. The aluminium block at the bottom has Peltier elements screwed to it, and by sending a current to this Peltier element, it can either cool or heat the aluminium block. If one side gets cool, the other side gets hot. And therefore it's slowing down the minute you start using it to cool, because the other side gets hot, and you need to cool that with ventilators, obviously. And you can see on the top there is a standard microtiter plate. With these machines you could do PCRs in 1.5 hours, 2.5 hours, maybe sometimes an hour. And then from this time on, these machines were introduced in the early 90s. From this time on, there has been a quest for ever faster solutions of PCR. So what came next was Carl Whitwer's invention, who started Idaho Technologies, and he was using glass capillaries. And inside the glass capillary you could put possibly 20 microliters of PCR mix, and the glass capillaries were hanging in a cavity, and they were thermostated by air. The air was heated by a light bulb, a halogen light bulb, and as we all know from our physics classes, light bulbs produce light, but only 1 or 2%. The rest is heat, so they are actually quite good heating elements. And then for cooling, a valve was opened and ambient air was streamed into the system to cool the glass capillaries. Later you will see that these glass capillaries share at least one problem with Eppendorf tubes. I will get to that a few slides down the presentation. Actually, this device with glass capillaries was the device that brought me into contact with PCR. At that moment, we were working on denaturing gradient gel electrophoresis. Many of you will remember that from the maybe mid-90s to 2008, 10. And we actually made a device where you can see a buffer bath here on the top, a buffer bath here on the bottom for electrophoresis purposes. Then there are glass capillaries here with a gradient gel inside, and on top of that gradient gel we were able to put PCR mix, and then in a system that roughly looked like this, here is a ventilator with a heating element. We could blow air around the system past the glass capillaries, and we could heat them and cool them to the correct temperatures to perform a PCR. And in this system we could perform a PCR in around 20 to 30 minutes, very similar to the Idaho Technologies system in the previous slides. And when the PCR was done, we simply switched on the electrophoresis voltage, and the samples we created in the PCR were run down the gel, and after running a camera system here that could move to 96 positions, took 96 pictures of the capillaries. And then in the next slide you can see here we highlighted one capillary, and here is a densitogram. So here you can see the peaks of the denaturing gradient gel electrophoresis very clearly, and we could use that to to detect mutations in human disease genes, and with possibly such a high reproducibility that from the pattern you see, you can pinpoint which mutation it is in a fragment. And then here we see this first derivative. So if you're at the top of a peak here, this goes through the zero. Okay, so that's enough about the past. Now, how did I get to this invention? Well, I was in a meeting speaking about detecting pathogens in cow milk. That's very important because cattle suffers a lot from other infections, and that can contaminate the milk with pathogens. And the sooner you can detect them, the better off you are. And it has to be, of course, a very fast test. So we were all in this meeting, and one of the participants came up with microfluidic chips that were around for some time then. And in these microfluidic chips, a very small amount of liquid is flushed through a labyrinth and meeting the several different temperatures needed to perform a PCR. Now, it has a number of advantages and a number of disadvantages. The big advantage, of course, is fast heat transfer. So with these chips, you are able to perform PCRs in minutes rather than hours. So that's the big advantage. One of the disadvantages is it's always a fixed number of cycles. You cannot vary the number of cycles. So if you want to do qPCR, real-time PCR, you are still at a fixed number of cycles. And it's a very small volume. If you want to do PCR in half a microliter, you need to pipette nanoliters. And many of you will be aware of the fact that pipetting nanoliters is not so simple. In fact, it can be very expensive to buy the equipment to pipette nanoliters. And on top of that, you know, after the PCR, if you want to process your sample further, you don't have a lot of it. So then I thought during this meeting, and I thought only, so I could just push back the words, because you know how it goes in meetings, you know, you think of something, you are participating, so you want to put it on the table. But I didn't. I could prevent myself from putting it on the table. So what if we could use something similar to a microfluidic chip, but normal volumes, at the same time mix the sample in every temperature zone, and not just move the sample, but also the enclosure. Then we would end up with a system that would have the same advantages as a microfluidic chip, but would not have the disadvantages. So here is the very first experimental setup. So let me see if I can, with the laser pointer, point out what it is. So here is an aluminium block, small, which can be kept at a constant temperature. Then just on top of that is a plastic isolating block, then there's an aluminium block again, then an isolating block, and then here should be a third aluminium block. On the rear side you can see an aluminium block on the top, then here in the middle one, and one at the bottom, and the one at the bottom still has the heating element and the PT100 in order to regulate the temperature. And then we made a plastic enclosure, so sort of a blister, thin polypropylene plastic with PCR mix inside, and fixed that to a sheet of the same plastic, so we could pull that vertically between the blocks to a position, and then one of these plastic screws we could use to push the blocks together and squeeze the blister a little bit. We put plastic screws in here because then you don't burn your hand when you push them. And the first experiment was together with Ronnie Kammerts, with whom I'm still working, and he was pulling the blister up and down and pushing the blocks, and I had a stopwatch and said next, 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 and we did 30 cycles in 10 minutes, and immediately we had a result. And not just, not just some result, but actually a quite perfect, clean result with a good yield. So then we thought, okay, we have something here. We need to make a product out of that. And so we did. We made this machine, and it had a polypropylene foil with 96 wells that we could fill with 96 PCR mixes and then close. So then we ended up with a plastic foil with 96 blisters filled with PCR mix. But we tried to make it small, so we took a quarter of a microtiter plate, we spaced the wells 4.5 millimeter, so we ended up with 16 times 6. That's a quarter of a microtiter plate. And we thought 16 times 6 is also 96, and you can still use multi-channel pipettes to load it in the direction of the 16 wells. You can load 1, 3, 5, 7, 9, etc. And then the next load you load 2, 4, 6, 8. And we thought, okay, that's good, that's cool. And with this system, we brought the PCR 30 cycles down to around 3 minutes, which was quite amazing. And then I met somebody who has been selling PCR machines for a long time. I think he sold his first PCR machine in 1985 or 1986. I don't know which one that was, but he'd been in the market for a long time. And he said, I like this and I'd love to work with you. His name is Simon Nicklin. But you've done one thing wrong. You made a beautifully fast PCR machine, but it's the wrong format. It has to be microtiter plate format. Okay. So we went back to the drawing board and we made this. Now here you see our microtiter plates. They are a frame which follows exactly the SBS standard. So this is the low frame that you all know from the 384 well microtiter plates. But we put a polypropylene foil on the top and then 96 wells. And on these plates, the ones that you see here, we put 96 wells of a maximum of 20 microliters in it. So you can load anywhere between 5 and 20 microliters. And those are actually quite normal volumes. And then we close it with a heat seal in a standard heat sealer with our special anvil. And then we put them in this machine. And the machine cycles the plate between zones. I will, in a few slides down, I will show you exactly how this cycling between zones work. So far, this is all pretty simple, right? So then we came to the GUI, the graphic user interface. Now, if you look at Pilche-based cyclers, many of them have large screens, more and more colors, and user interfaces with more and more possibilities. We didn't want to do that. I tried to keep it as simple as possible. So what do you need to do if you program a protocol? You need to feed it 3 temperatures: denaturing, annealing, and extension. Then you need to say how many seconds initial denaturing time you need. Then how many seconds denaturing you want in each cycle. Then how long you want to anneal in each cycle. And then how long you want to extend in each cycle. And then if you want a final extension, you can put it in here. And then here is the number of cycles. And that's basically, apart from a touchdown, step down, or a few tricks that you need, or that you might need, that's basically all you need. So if you fill in your figures here, and you say save, you can save it under a name, but if you say run, it will start to run immediately. Now I would like to go a little bit to the difference between Eppendorf tubes and our plate. So this is a standard Eppendorf tube, and the thickness of the wall of the polypropylene tube, if you have a thin wall tube, is around 200 micrometers. Now, this is a corner of our plate, and you can see here 1, 2, 3, a fourth well, 1, 2, 3, 4 wells. The wall of the well has a thickness of around 40 micrometers. So that's 5 times thinner than an Eppendorf tube. So that should help speed things up. You would think, but that's not all you need. Because not only is polypropylene a good isolator, still standing liquid as still standing air is also a good isolator. You can think of a thermos bottle which you use to keep your drink cold or hot. That's a double-walled bottle, and between the walls is air. If the air is not moving, it's a very good isolator. If there would be liquid, it wouldn't be as good an isolator, but if it's not moving, it would still be a pretty good isolator. So if you look at the standard Pilche block thermocycler with an Eppendorf tube, then you take it from denaturing temperature to annealing temperature. That's the biggest distance. So let's say it's going from 95 to 60 degrees. And you look from the top, then you will see a liquid surface inside the Eppendorf tube, which looks a little bit like this. So on the outside, the liquid will be close to 60 degrees because the block is 60 degrees now. But then if you go to the middle, it gets hotter and hotter and hotter. So here right in the middle, it may not be 95 degrees anymore, but maybe it's 75 or 70 or 80. Do we know? No, we don't. Nobody measured this, and it's also very difficult to measure because any thermometer has its own thermal mass and influences the measurement. So it's difficult. But what you can measure with a very small PT100 is how long it takes for the liquid in the middle to be also 60 degrees. And that is a little bit depending on the volume in your Eppendorf tube, because as you put more volume in it, the radius of the circle of the cross section of the liquid increases, but it's around 20 seconds. So that means if you think about it, you lose 20 seconds every time you change the temperature. And maybe from annealing to extension to denaturing, a little bit less, but still, 10 seconds, so 40 seconds per cycle, you lose. Times 30, that's a lot of time. So how do we do it then? We have 6 aluminium blocks depicted here on the top. So suppose this is the denaturing, then here I have 2 blocks at the same temperature. They are both 95, 98 degrees, whatever denaturing temperature you choose. In the middle I have the extension zone, so these 2 blocks are both the same temperature. They are both 72 or 74 or 75 degrees. And these 2 blocks are both at annealing temperature. Now, if this block is now at extension temperature, it means it just came from the annealing zone, so the blocks open, and the plate which you see here with the foil with wells is moved between the blocks of the extension zone, and then the blocks close. And you can see that the wells are actually smaller now than in the top picture. And that's because we squeeze them, we put pressure on them. And by doing this, we mix the liquid inside the blisters, inside the wells. And by mixing this liquid, we make sure that 1, we have a very good surface to volume ratio because we flattened it, but 2, because we mix the liquid actively, all of the liquid immediately is at the correct temperature. meaning we take out the ramping time that you need in a PCR. Now, the next slide is a picture of the machine which you saw in the first slide, but with clear size, so you can look inside. Now here you see the top block of the annealing zone, and here the bottom block of the annealing zone. It's isolated in this one. This is the extension zone, top block, bottom block, and this is the denaturing zone, top block, bottom block, also isolated. And then between the blocks we can move the plate, which is loaded here in this zone, is moving on a belt all the way to the denaturing zone, and then the blocks close. I would like to show you a little short movie which shows 3 cycles of the whole process. As you can see, now it's here, it's an extension zone, it's in the denaturing zone. It's in the annealing zone, extension zone, denaturing zone. And this is really how fast it goes. I'll let it run until it's finished. And it stops in the extension zone here. It's obvious we stopped the movie, but it will continue 30 cycles, 35 cycles, 25 cycles, whatever you programmed. Now, as with any system, you need some accessories. There are a few things that might raise questions. So if your wells are 40 micrometers thick, only 40 micrometers of polypropylene, think about it, that's really very thin. So if you load those, you could stick through the bottom. Well, we thought about this. This is a pipetting anvil. It's made of a plastic called Delrin, and it has 96 wells which precisely support the bottom of the wells of the plate. And in this way, it's impossible to stick through the bottom because you will hit the bottom of this pipetting anvil. So the plates fit on it like this. Here you have 2 cut-off sides. The pipetting anvil has the same 2 cut-off sides. And in the next slide, you can see that it's very easy to fill with an 8-channel pipette. And then the first thing people ask, okay, so these wells are not so deep. That's true, they are not so deep. But still the meniscus, the top of the liquid, is below the surface of the well if you put in 20 microliters. Good. So now you have to be very careful with this plate, of course, because otherwise it will spill over and it will contaminate the next well. You would think that, but here physics is helping us. The surface tension of the liquid keeps the liquid together, and the attraction between the polypropylene and the liquid keeps it in the plate. So you can actually take this plate, keep it upside down, and nothing will fall out, nothing will happen. Meaning that you could use it, for example, with the acoustic pipetters that people start to use now. They shoot up a little bit of liquid to a microtiter plate, and a little bit of liquid stays in the microtiter plate, which is upside down above the acoustic pipetter. This plate would also work. And then you need to seal it, because we put pressure on it, so if you leave it like this, all your liquid will be evaporated. If you try to use a sticky seal, it's fine for a normal plate, not strong enough here. We put pressure on it, the liquid will evaporate. So here we have a second anvil that you need to use with the system. It has 96 individual cylinders, and each cylinder supports the foil around one well. Next slide, you can see that we put it in a sealer, in a standard sealer. It could be any sealer. This is a K-Bio one, the 40 tubes work, any sealer will work. It's filled with liquid. We put a heat seal on it, close it, and when it comes out, for example 2 seconds at 165 degrees, you can see that it's sealed to the plate, actually around each well. And if you take the plate off the anvil and look underneath, you can see 96 perfectly round blisters filled with liquid PCR mix, usually. And this plate can be put in our system. Just load it here, close the lid, push run, and it will start. So here is the slide that I've shown before. You can put all your data here, 3 temperatures, 3 times, initial denaturing, final extension, number of cycles. You can see that we put initial denaturing 30 seconds here. This is an immuno deactivated hot start. The chemical deactivated hot starts need 15 minutes of initial denaturing. Well, if you're going to use 15 minutes of initial denaturing, you don't have a lot of chance to do a 10-minute PCR, obviously, because you're already 5 minutes short. And then we also give you the possibility to do a touchdown. You can see that you can divide your cycles into 5 groups. In this case, cycle 1 to 5. If I go 1 slide back, you can see that the annealing temperature is 60. Cycle 1 to 5, we take down 0.1 of a degree per cycle. So that's half a degree in total. Then cycle 6 to 10, 0.2 degrees per cycle. So that's 1 degree. And then 11 to 15, 0.3 degrees per cycle. That's another 1.5 degrees. So in total, we take out 3 degrees. So we end up with 57 degrees annealing time after 15 cycles, and then cycle 16 to 30 will automatically be done at that 57 degrees. And of course you can also do step down, where you simply say cycle 1 up to and including 5, I want to do at 60 degrees, the next 5 I want to do at 58 degrees, the next 5 I want to do at 56, and the rest at 54. Again, this is designed to be easy to program. And then we have an advanced mode. This one is actually showing the 2-minute protocol. It was a 100 base pair fragment, which we did in 2 minutes in 30 cycles. So denaturing temperature, you can see that we choose a little bit higher, 98 degrees. The higher the temperature, if your enzyme can stand it, use it. Denaturing goes faster. Then annealing 60, extension 75 was optimal for this enzyme. 10 seconds initial denaturing. Obviously, if you don't use a hot start, you can save 10 seconds here. Then I have 2 seconds per temperature for 5 cycles. Because after 5 cycles, I have a little bit of product. So I don't need to denature the whole template anymore, but just the product that I made. I can do that in half a second. I don't need to anneal to the whole template. I need to anneal to the fragments I made. I can do that faster. And also the enzyme finds its position faster on short fragments, on the 100 base pair fragments, than on the whole template. So again, half a second. And then we end up with 2 minutes. And then as you choose options in the software, you are getting screens deeper and deeper into the graphic user interface. If you want to go back, simply push this one and it takes you 1 screen back, always. And it looks like this if it's running. This one has 95 degrees, 50 degrees, 72 degrees, and with this ring you can see that it is now in the extension zone. The plate is now in the extension zone. If you press the red button, it will stop, the plate will come out, and the thermostation of the aluminium blocks, of the 6 aluminium blocks, will stop and the temperatures will start to drop. If you open the lid, it will only pause. You close the lid, it will continue where it left off. And then this you will recognize from an earlier slide as the pipetting anvil, and we also use that to retrieve the PCR products from the plate. And we do that using this specific tip. This is still a polypropylene pipette tip, but as you can see here at the end, it's cut off at 30 degrees. So it's really sharp. It's sharp like a needle. And what we do with this is simply stick through the seal, the heat seal. And the heat seal is, in this case, a sheet of thin aluminium on the top side. So you can use heat to heat it up without sticking to the heating block. And on the other side, the plate side, is a thin layer of polypropylene. There are also clear seals that we can use. They are polyester on the top side and polypropylene on the bottom side. These tips will also stick through polyester seals. And then obviously you can also do that using an 8-channel pipette, and you can see that here it empties 8 wells at the same time. Now then here is an interesting let's say, result of the design of the machine. The 6 blocks that we have, the bottom blocks on the top and the top blocks on the bottom of the blocks, have flat surfaces. So a top block has a flat surface on the bottom, and a bottom block has a flat surface on the top. So the wells are met when the blocks squeeze together, are met by flat surfaces. That means I don't need to put in a specific sort of plate. On the left here, the blue one, you can see a plate with 96 wells, which holds between 5 and 20 microliters. On the right, this is what you are used to also now. On the right, you see a 384 well plate. This has 384 wells, which holds between 1 and 5 microliters, maximum 5. This you also know. This you can stick in the same machine without changing anything. It will accept both plates and do a PCR in both plates. And now what's interesting here is a plate that doesn't exist currently. It's 96 wells, but they are small wells. So you can use the wells for 1 up to maximum 5 microliters. So now you have a 96 well plate, which you can use for 1 or 2 microliter PCRs. And that wasn't available previously. Then across the block uniformity, our blocks need to be kept at constant temperature. We are not cooling or heating the blocks. We are simply keeping them at denaturing, extension and annealing temperature. And they are isolated on the top for the top blocks, on the bottom for the bottom blocks, and all around. So they're not losing a lot of energy. So our across the block uniformity is exceptional. And I can show that here. We took a plate and loaded PCR mix at 14 different positions all across the plate and put it on a gel. And well, I think this is pretty uniform. Good. Then what can it do? We went to the University of Rotterdam, Erasmus University, Clinical Genetics Department in the Netherlands, and they are doing their BRCA1 diagnostics using PCR Sanger sequencing, followed by Sanger sequencing. And the demand was, we need to be able to do all 29 fragments under the same protocol. Would we have been able to use different protocols? Then probably we could have gone a little bit faster. But anyway, you know, we ended up with doing all 29 in 9 minutes and 40 seconds, 30 cycles, longest fragments up to around 800 base pairs. They look okay on the gel, and after doing the PCR and then the consecutive sequencing reaction and loading them on a sequencer, the sequence data looks perfectly readable. All 29 fragments were sequenced like this, and all were, of course, without mistakes, because in the end, it's the fastest thermocycler in the world on a microtiter format, but it's still a thermocycler. And then here you can see that we can do 30 cycles, and I said 2 minutes. That wasn't true. It's actually 1 minute 59 seconds. And I think for mechanical reasons, the limit for this machine will be between a minute and a minute and a half. So what we did here was 5 cycles, 2 seconds per temperature, and then 25 cycles, half second per temperature. And these are the results. 100 base pairs. And then here I show a plate which holds a fluorescently labeled sample in one of the wells, and which indicates towards where we want to go next year. So now we have the endpoint PCR cycler out, it's available, people are using it, they're getting good results. But we want to come with a qPCR machine next year, which can do qPCR in a matter of, well, minutes also. And then I would like to thank Canon BioMedical for sponsoring this webinar. It's a great opportunity to speak to so many people at the same time. So thank you for that. And Canon BioMedical are also our distributor for NEXTGENPCR products in the US and Canada. And again, if you want to visit our site, nextgenpcr.com. Thank you all for your attention.
Okay, so a huge thanks, Gert, for that very thought-provoking presentation. Always fascinating to hear the genesis of these technologies, and great to hear the process of how you invented this technology. So thank you very much for a very, very engaging and inspirational talk. We're ready now to start our Q&A session. I'm glad to say we've had some good questions are being pinged through, which is great. Always a good sign of a good talk, I think, when plenty of people have questions. So we'll start to go through some of these. We'll try and get through as many as we can, given the time constraints. So the first one, Gert, I have, if you're able to answer, is what is the largest base pair fragment you have been able to successfully amplify?
Well, actually, that was during a demo in a lab in the UK, and it was 1500 base pairs, so 1.5 kb, in 13 minutes. But because it's a thermocycler, there is no reason it couldn't amplify 10 kb or 20 kb. But then, obviously, you still gain the 40 minutes per amplification in time, so it will be faster. But the relative gain will be less because you need longer extension times. But 1500 is what we've been doing up to now.
Okay, good man, thank you. Next one here, is there a specific chemistry that needs to be used with the instrument?
No, actually not. It's the standard PCR chemistry that you use. But if you want to go fast, you need to use an enzyme which doesn't use a lot of time to activate. So if you want to use a hot start, use a hot start that can activate in 10 seconds or so. I know that manufacturers say that you need 30 seconds or 60 seconds, but I think they want to stay on the safe side. Our experience is that usually 10 seconds is enough to activate the enzyme. So, and then it's just a matter of the standard buffers that you get from your manufacturer or that you make yourself and you're used to using, and that's all. That's it.
Okay, brilliant. Thank you. Another one here. Given that common sensitivity of qPCR systems requires enrichment of food sample for pathogen detection, which is a big time bottleneck, even higher than the PCR step itself, how the sensitivity of your system compares to other qPCR systems?
Yeah, because it's still a PCR system, it will be around the same.
Right. Okay.
And the sensitivity, I think we can do a little bit better because of the design of our plates, but I'm not willing to disclose a lot about it because we are still in the process of selecting everything and also in the process of patenting everything for the
Right.
qPCR. But the special shape and form of our plates give us a little bit or a few extra possibilities to increase the sensitivity a little bit. But very often it's better to look at the front side, so the step before the PCR, to increase the sensitivity. And for example, what a group in Groningen has been doing is detect UPEC, urinary tract pathogenic E. coli, for urinary tract infections. And they take urine and simply press it through a filter, and then wash the filter with a little bit of water and use that water as a template. So maybe with food, after processing it in a food processor, you could do something similar, but I'm not sure.
Okay. All right. No, well, thank you for that. Next one here. Can we only use plates on these machines? Can we use individual tubes or 8-tube strips?
No, you can use only plates because we need the frame in a carriage that shops the plate from the back of the machine to the front of the machine and back. So no, you cannot use individual tubes or 8-tube strips, but we are aware of the fact that not everybody always use a 96 well plate. So we are going to come out with a plate that has only 8 and a plate that has only 24 wells. And therefore you don't waste a whole plate then. And they will obviously be more affordable to buy than the 96 well plates.
Okay.
So we will accommodate those users.
Okay, thanks, Gert. Another one here. Maximum volume, is that only 20 microliters?
For the 96 well plates, it is. So for the plates that we have commercially available, it is. But actually, the maximum volume for a full plate, so for the 96 wells combined, is 2 ml approximately. So 96 times 20 microliters is approximately 2 milliliters.
Right.
So we could, in theory, you could make plates with 4 times half a milliliter if it had to be used for production of DNA vaccines, for example. We've been thinking about that, but currently we're not going that way. So until now, yes, the maximum volume is 20 microliters.
Okay, thanks, Gert. Can a single heat block apply gradient of temperature?
No, we cannot. So that's a simple question, simple answer.
Simple answer. Okay, all right, thank you.
Well, there is a little bit more to say about it, because usually you do a gradient temperature across a block to be able to see which temperature is best to use in your assay. Mm-hmm. But because we do the assays so quickly, with doing 4 PCRs, you have the same answer, and in shorter time than on a gradient block.
Okay, all right, brilliant. How do you recommend reading post-PCR containing fluorescent hydrolysis probes? This unit does not appear to capture real-time images or flaws.
You'd have to wait for the real-time PCR units, but otherwise, one of the possibilities is obviously pipette them in a normal plate and stick them in a qPCR machine that you have in your lab. or in a high resolution melting reader or other fluorescent readers that are around. And since our plates are standard SBS format microtiter plates, they should fit.
Okay, brilliant. Is it possible to skip extension step?
Yes, this is probably meant to ask, can you do a 2-temperature PCR? And the answer is yes, you can. If you select 0 seconds for the extension step, it doesn't do the extension step. It goes between denaturing and annealing, and then it skips the extension step. I have to add that in our experience, because we virtually don't need any time to go from one temperature to the other, most assays are quicker using 3 temperatures.
Right, okay. When the wells are pressed, does it squeeze the wells too? I've got here.
Yes, it really squeezes the wells. So they are going for example, from 5 millimeters high to 2 millimeters high. And this is why the liquid inside the well is mixed. And this is one of the reasons why there is immediate heat transfer. So they reach the wells reach the next temperature immediately. Yeah, so yes, we do.
Okay.
And they come back after you release them. They come back to their original form because they are closed, so they have a higher pressure inside.
Okay, all right. Thanks, Gert. How much temperature drift do you see with the cooler plate moving into the heated zone and the hot plate moving into the cool zone?
Yeah, that's an interesting question, and that's something that we anticipated with the design of the machine. So the weight of the aluminium blocks is calculated to allow a 0.1 degree drift when a full plate of 96 wells arrives from 98 degrees denaturing to a 48 degrees annealing. So with a 50-degree difference, the drift is 0.1 degree. So that's actually nothing to worry about.
Okay, all right, thanks, Gert. And we've got here, does the sealer come along with the PCR instrument?
Yeah, we can do if you pay extra. Yeah, it doesn't. You can use your own sealer if you have one. We will make an adapter if necessary for our anvil. Usually it's not necessary, usually it just fits. So you can use your own, but if you don't have one, we'll be happy to sell you one, or you can buy one from anybody else. And they are, the different brands, the semi-automatics are comparable in price, I would say.
Okay, brilliant. Is it, let me just see here if we've got time for a couple more. Let's have a look. Has any work been done for multiplex PCR?
Yes, of course. Many of the forensics kits, for example, work with 12-plex or 24-plex. And some of the older kits, which try to use the fact that normal PCR machines can do very slow ramping, take much more optimizing, but the modern kits, they should normally work. Yeah, it takes a little bit of optimizing, but not too much. They work.
Okay, brilliant. And is there an easy way whereby we can shift the assay from normal PCR to NG PCR?
Yes, there is. The first thing you do is simply copy your protocol. So you have timings for different temperatures. You copy those temperatures, you copy those timings, and to our machine, you stick a plate in and you have a result. Usually. 99.9% of the cases, you have a result.
Okay.
But you have it 40 minutes quicker. 20 to 40 minutes quicker. And then you can start to squeeze. Because of the slide that I showed, with the Eppendorf tube, which has rings of fluid of different temperature. We don't have that. So this is why you can then nip time off the different timings for the different zones. So the one to start with is the denaturing. This can usually be taken down to 10 seconds. Then in our experience, the annealing zone is the one to leave alone and start squeezing the extension zone first, because most enzymes are faster than manufacturers tell you. But then again, I had a discussion with Carl Whittler just last week, and he told me that in his experience, the extension zone is the one to leave alone, and you can squeeze on the timing for the annealing zone. And of course, if you want to do that, it would increase the concentration of primers, then the annealing zone is a very, very good candidate for squeezing down the time.
Okay, brilliant, thanks, Gert. Are you planning to make it real time?
Yes, we are working on it, and in the course of next year, we should have one. Yeah.
Okay. The next question says, great talk. Was the air temperature in the machine above ambient, or is it preheated?
No, the blocks are preheated. So if you switch on the machine, the blocks start to heat up to certain temperatures. If you have different temperatures, you need to program those. Wait a little bit until the blocks are at that temperature, but that usually doesn't take more time than it takes you to prepare your PCR mix. So if you do that first, make your PCR mix, then it'll be ready for you. And the ambient temperature in the machine in the end gets above ambient because it has 6 hot blocks in it, and although they are isolated, the temperature goes up a little bit, but I think it's around 40-45 degrees, not very hot.
Okay. Which Taq polymerase was used to amplify?
The 2 experiments that I showed, the breast cancer 1 and the 2-minute PCR, have both been done with kappa-2G fast, but we also have very good results with a number of other enzymes. We used, for example, platinum Taq, QuantaBio, AccuStar, titanium Taq, SilverStar, Fusion, Dream Taq, Go Taq. The only one we had a little bit trouble with was the AmpliTaq Gold, which still many people use, and it uses 15 minutes to activate. So then you cannot do PCRs that are really fast because you lose the 15 minutes in the beginning.
Right, okay, which pretty much I think answers the next question. The next question basically had said, are there any particular vendors, polymerases that work best with the instrument? But I think you've made
most of them will. Yeah, and we tried quite a vast number of other enzymes also with demos at customer sites, and we hardly ever saw any problem. Only if you have a Taq that needs 15 minutes denaturing, you lose time.
Right, okay. And is it easy to unseal the plates?
To unseal?
Unseal, yeah.
No, you cannot unseal them because it's a very thin foil. So if you would do that, your sample would fly all over the place. So what you do is pierce the seal and then take out your sample. So if you want to unseal them, add something, reseal them, that's one of the things we cannot do.
Okay. I'm sorry. I mean, I know I'm just going to try and get the last 2 or 3. I know you've been very good, Gert, so sorry for keeping you. I know you probably want to get home for your dinner, but does 3-step cycling get between denaturing temp and annealing temp faster than just a 2-step protocol?
Can you repeat the question?
Sorry, yes, indeed. So does 3-step cycling get between denaturing temp and annealing temperature faster than just a 2-step protocol?
No, there's no difference. No, because the temperature only starts changing once the plate is in the next zone and the block starts closing. but to give you an idea about how fast the plate is really moving, so the furthest it can move is between denaturing and annealing, and that horizontal movement takes 0.1 second.
Right, okay. And we're coming down to the last couple here. Can we use all types of tack? I think we've probably...
Yeah.
Yeah, okay. And then, very nice talk. Can we revisit this webinar later? I can answer that. Yes, of course you can. It will be posted on the website for 6 months on the Journal of Precision Medicine website, and also you'll be able to download it, I'm sure, at the Canon website too. And he's asked whether you can please share the slides with them, but I'll leave that for you to decide with.
We can share the slides. I have no problem.
Okay. All right. Fantastic. Well, look, we could probably spend all afternoon here. I think it is absolutely. It's an indicative of a brilliant talk when you get this many questions. I've not had this many questions before, if I'm honest. This is great. But
I'm looking at the questions now. I can also see them. So can I just pick two more and answer them?
Of course you can. No, of course you can. Look, like, you know, as long as you want. So I'm not going to hold you back. You just go ahead.
There's one very practical question here. How much do the plates cost? Well, they won't cost more than your normal plates that you are using now. It's in the same price range, so it doesn't make your experiments more expensive. And the other one I would like to say something about is, have you tested this with library prep kits for NGS? The answer is yes. We've tried that, not very often, but we've tried it on a few occasions during demos in the United States and in the United Kingdom. And on one occasion, we got the result which was very, very comparable to a standard cycler. And in the other case, it looked like we got a little bit more specific result. But we are going to do more work on this because we feel that we should be more specific as we change the temperature instantly, and we don't end up with tubes with liquid of many different temperatures. So yeah, that's a very interesting part of the applications for us. Yeah.
Okay. Well, look, I'll tell you what we'll do. If any of those questions haven't been answered, then we'll make sure that we will, and we'll make sure that Gert and Canon obviously circle back with all of those questions. So we'll endeavour to make sure all of those are answered. And of course, you're able to contact Gert or Canon obviously directly, and we can help facilitate that if you have any problems. So look, as I said, we could go on and on, but I think it's been a fantastic talk. It's clearly, that's the consensus. We can see that from all those fantastic questions. Look, I mean, all it leaves me to say is to finalise proceedings by thanking all of the registrants, indeed, for joining the webinar today, and a special thanks to Gert and for an excellent talk, and obviously Canon BioMedical for the support in making this webinar available today. It has been recorded, of course, and we will be posting on the website, as I said, for 6 months, and it will be accessible on Canon BioMedical's website too. So I wish you all a very pleasant rest of day, wherever you are, and we look forward to welcoming you back for another Journal of Precision Medicine webinar very, very soon. So it just leaves me to thank you all and catch up in the near future.
Thank you very much.
Thank you, Gert.